welcome everyone. Glad you could be here in person, and for those that are in different parts of campus or in remote parts, glad to have you as well. So we're delighted and honored to have Hamed Ektiari here visiting us, coming from University of Minnesota. Hamed is a very unique individual. So trained in Iran, has an medical training and PhD training there, and then came to the United States. He worked in Tulsa, Oklahoma at the Laureate Institute for Brain Research for several years, and essentially established his own lab and started racking up publications and really garnering recognition nationally and internationally for his role in neuromodulation and addiction, especially, and that's sort of the specialty niche. And so we're hosting him here. He's actually a fourth-year resident at the University of Minnesota right now in psychiatry. And so I think he probably holds the record for the most publications from a medical resident in history. If not, I would put it up against anyone in that regard. So anyway, to have Hamed here and visiting us is a fantastic thing. We're hoping to be able to recruit him and to work in the TMS and neurotherapeutics group, and also with our addiction group, and he'd be a fantastic bridge for us in that regard. So anyway, without further ado, we'll turn it over to Hamed, and delighted to have you. And also...
Okay, hello everyone. It's just delightful to be here, and thank you very much for the invitation. It's good to see friends and colleagues here, and many thanks to Dr. Brown, and I'm going to use that, sure. And kind of thank you very much, Josh, for putting this together, and the energy and positive inspiration. I know Josh for quite a while. I've always been inspired by his energy and kind of positive attitude and support for junior colleagues. And thank you very much, Josh, for kind of that attitude. And I really appreciate that. So as Josh mentioned, I'm very interested to basically addiction and addiction psychiatry. Meanwhile, I have been kind of doing most of my residency doing TRD and seeing people with kind of having comorbid anxiety, depression in the context of addiction, and trying to put those things together. But what I'm going to present today is just mainly about the ideas of how neuromodulation could reshape the future of addiction treatment and potentials that we have in this area, and kind of the specific things I'm working for this kind of line of activities. I do not have any conflict of interest. And before getting started, I would like to thank my colleagues and collaborators, especially Martin Paulus. I was his postdoc for 2 years, and then a colleague for several years. And I really learned a lot from him and many other colleagues at Laureate Institute for Brain Research. And also my kind of team of supervisors and colleagues at the University of Minnesota. I'm enjoying working with them, and they have been very generous to me for whatever I'm presenting today. And without their support, it was not possible to be a psychiatry resident and have some like 4 postdocs in the lab and 2 ongoing clinical trials. So all those things have been kind of possible because of their support. And I really appreciate what they have done for me. And my lab members that, I mean, whatever I'm presenting here are basically what they are doing. And I'm just delightful to be working with kind of energetic young colleagues that are brilliant minds and the kind of the future of the field. So let me talk about neuromodulation. It's not necessary for me here to just give you an idea about what are different technologies that we have for neuromodulation. So there are lots of different opportunities. There are lots of different technologies that people are using for neuromodulation. Specifically for the field of addiction, we have been kind of using electrical stimulation and magnetic stimulation, different forms, to be able to kind of modulate activities related to drug addiction to be able to develop treatments. But when we think about kind of TAS and TMS as technologies, the number of publications in the field is just growing up over years, which is impressive to see in terms of right now we have over 1,500 up to something like 2,000 publications, specifically original publications with TMS. And the field is just growing, and you see the progress over the last, let's say, 20, 25, 30 years. When we talk about addiction, just one slide about the prevalence of addiction. What is the percent of the people who suffer drug addiction in the past year in the US? Anybody has any idea? The percent? And 25 is, yeah. Okay. It means that from 4 people, 1 would have kind of addiction. I mean, we have a specific definition for substance use disorder, but just kind of within the definition of substance use disorder, we have 15% within the last year who suffer from drug addiction. And the large portion is alcohol, but also illicit substance use disorder is around 20 million people here in the US. So the number is huge and the burden is huge. And you know that for many of those drug addiction kind of types, we actually do not have any FDA-approved medication. So there are lots of challenges with methamphetamine, with cannabis, with cocaine, with all those kind of, other than opioid use disorder and alcohol to some extent. Even when you kind of work in the areas of alcohol and opioid disorder, you realize that the number of options that you want to provide for people who are suffering from those disorders is really limited. So if you go beyond naltrexone or compressate and just kind of on-tap use, I mean, you do not have that many options compared to what you have, for example, when you work in TRD clinic and you have a longer list of different options that you have. Then when you work in the field of addiction, you realize your hands are really tied. People started to use neuromodulation for addiction, both magnetic stimulation and electrical stimulation from years ago. Do you know what was the first study in the field of addiction with neuromodulation? So first study with TMS and cannabis, 2018. So the first TMS study with cannabis use disorder happened 2018, which is pretty surprising, right? It's just a few years ago. And then first study with TMS was 2003, first TMS cocaine was 2007, and then alcohol 2010, first TDC study 2008. So field is pretty new. So the number of studies that we have, most of them basically happened within the last 10 years. And as you can see here, field has recently started to develop literature and having an idea about which of those kind of pathways are going to work. First TMS study on opioid, 2016, just basically 8 years ago. And then there are lots of kind of developments. First FDA approval or FDA clearance for smoking happened 2021. And first individualized TMS study, 2023. First high definition TDCS study happened in 2022. So many of those studies are pretty new compared to many other areas of neuromodulation compared to what we had in depression or other areas. Many of those studies, those kind of things are pretty new. And then how many published trials we already have with TES and TMS for addiction by the end of 2024? Any estimate? Published randomized clinical trials in the field of addiction by the end of 2024. 25, okay. So we have 242 published trials. Okay. And this is something I basically updated in my kind of flight to here. So I was just today in the morning, I basically updated this slide, putting together the kind of the new data. I'm tracking all the publications and sort of I'm doing some of a living systematic review of whatever people are doing, just tracking whoever is making a new publication, basically add that to it and rerun my R code, just making sure I have all the figures updated. So that is a sort of an obsession that I have, just making sure I'm tracking whatever people are doing in the field and enjoying the progress of the field. As you can see here, these are the TMS studies, these are the TES studies. Nicotine is the highest number, of course. And last year we had 14 clinical trials in the field of nicotine use disorder. Of course, afterwards is alcohol, amphetamines, cocaine, opioid, and then TMS, TES, as you can see here, the number is just increasing over time, especially with TMS. There is sort of like a consistent increase over the time during the last couple of years. So to 24 TMS studies in 2024 in the field of addiction. So the field is progressing. And if I put kind of the studies that they provided enough data that you can actually do a meta-analysis on the data that they provided, okay? And with two main measures, craving and consumption. And of course, consumption is a more sort of conservative outcome measure compared to craving. And as you can see here, these are the kind of reddish numbers are for TES, the bluish are for TMS. We have something like 69 trials with craving and 45 trials with consumption. We have enough data that you can actually run a reasonable meta-analysis. Please. Exactly. And basically putting them all together at the kind of same kind of pool. And the effect is overall significant. The effect size is something like a moderate effect size, small to moderate effect size. But as you can imagine, the effect size for craving is a little bit larger compared to consumption, which is a more conservative measure of what we have. But the overall effect is significant. But the challenging part is those trials actually targeted many different areas. So when you run the meta-analysis, you see the heterogeneity level is really high. People have really kind of different outcomes. And people have tried multiple different areas in the brain, which makes things a little bit complex in terms of any sort of conclusion from what people have been doing. And even when you go to the details, for example, when you go to, the most frequent area, as you can imagine, is DLPFC, of course. People target DLPFC more frequent than any other area. But if you go for DLPFC, you realize within DLPFC, people have used different ways of targeting DLPFC, EEG system, 5 centimeter rule, 6 centimeter rule, MRI guided, beam, 5.5. There are different ways of even defining DLPFC, okay? And most of them are basically skull-based measures, EEG-based measures. And based on the literature that we know from basically depression, those measures of targeting, skull-based targeting, EEG-based targeting, they're not that much accurate. They're not that much precise. Then kind of using structural imaging and targeting based on structural imaging could be more precise, but probably not the right area, not very much accurate. And then the idea of how we might be able to find the better sort of biomarkers using functional imaging, whether it's fMRI or PET, or connectivities, we have experts here who are actually doing this job with more high accuracy and high kind of precision. And also at the end of the day, it's just basically thinking about how we can do that in a personalized way, right? Targeting the personalized targets. And we have what we call group level target optimization and individual level target optimization. So in these two levels, whether we are optimizing things in the individual level or we are optimizing things at the group level, these are the two levels of optimization. And in the rest of the talk, I'm gonna kind of give you a little bit of idea about what we are doing in the same line of work in terms of how we might be able to do addiction, neuromodulation in more precise and accurate way, and also in a kind of individualized or optimized way. Okay, so when we talk about depression targeting biomarker, there are different discussions about what is the best biomarker target. The idea of basically targeting the DLPFC connection with subgenuous cingulate cortisone is involved. It's one of the kind of the key figures kind of bringing that idea. But the question is, what could be the biomarker for addiction? What would be the right area to target for addiction? And that's a question for us in terms of what would be the best kind of targeting biomarker, okay? So there's a wide range of different areas that people discuss in terms of what would be an area for kind of neuromodulation for addiction. As you can see here, the idea of engaging executive control network, engaging default mode network, saliency network, the connection between different areas. So there are multiple potential biomarkers that could be targeted in the field of addiction. And just giving you a sense about one of those in terms of engaging frontoparietal network, including DLPFC and parietal cortex, is an area that we have been interested to. I already have kind of a finished trial on using transcranial alternating current stimulation with what we call frontoparietal synchronization. It means that we try to engage frontoparietal cortex, frontoparietal nodes together with oscillating them in the same sort of frequency, and how we might be able to do what we call frontoparietal synchronization and engaging them with the idea of extending basically top-down regulation over subcortical areas. So to be able to do that, so the initial idea was, okay, when we want to kind of engage frontoparietal network, let's go for F3, P3, F4, P4 stimulation. So that was the kind of most traditional way of considering frontoparietal network. Okay, so we started with F3, putting the kind of the high definition electrodes on F3, and then putting the parietal node on P3, and defining what will be the node, and then measuring how we can have this network being engaged. And when we talk about drug craving, we know that in drug craving, frontoparietal network should be engaged, because when people are exposed to drug-related cues and they have craving, they start to engage their executive control when they are in treatment. They want to basically manage their craving, so the frontoparietal network should be engaged. But putting those nodes as kind of areas for connection or measurement of frontoparietal engagement, we realized with considering those 2 points, we did not have any frontoparietal engagement. So frontal are connected to each other in the context of task-based connectivity with drug cue reactivity. But frontoparietal, they are not engaged. So we're thinking, oh, we are running a trial in this area, it just doesn't look good that they are actually not connected when people are seeing drug-related cues in the context of task-based connectivity. So we started thinking about, okay, what is the problem? So when we put the seed over F3 and F4, we realized, yes, we have a node of connectivity in parietal cortex, but the node is actually not under P3 or P4. So we realized that the node is actually under CP5 and CP6. And if we change the target from P3, P4 to CP5, CP6, then we will have the engagement of CP5, CP6. So we realize that when we talk about frontoparietal network, there are lots of different frontoparietal networks, and which one is actually the frontoparietal network that we are interested to engage in what we are running. So beyond that, we realize that when we want to engage the network, if we want to measure the amount of electricity that we have in two nodes, when we put electrodes on F3 and P3, and the classic montage that we have been initially thinking about, for 2 milliamp on frontal, 2 milliamp on parietal, this is the distribution of electrical current in 60 methamphetamine users on frontal and parietal side, which is kind of has the same sort of distribution across different participants that we had. But when we changed it to another level of putting F3 CP5 as the nodes, we realized that the CP5 node is receiving higher level of electrical current. So there is no balance in terms of the level of electricity that we put in these two nodes. Maybe because of the difference in the skull thickness and the kind of CP5 area compared to P3 and P4 area, but the distribution would be different. So we realized if you want to actually have the balanced interaction between these two nodes, we need to change the level of electricity that we put on the parietal node, putting that something like a 1.2 milliamp compared to 2 milliamp that we had before, to be able to have a balanced level of electricity on both sides. So this is what we call group level optimization. So we have not been optimizing the dose in an individual level. We are optimizing the dose in a group level. And the protocol is going to be same for every, entire group. But the average of the group would be kind of equal in both nodes. So that is a kind of a sample of how we do group level optimization. Okay, does it make sense? Okay. So in this study, we are actually doing both group level optimization for target and also the dose. And we are optimizing the target in both group, in group, and also dose in group. We published the results in brain stimulations of the protocol and the pipeline, and the process is available for those who are interested. And now we are basically putting that to another level in terms of doing stimulation inside the scanner while we are recording fMRI. And this is what we call concurrent TES fMRI. To be able to do the stimulation while we are recording, to be able to see if we can actually measure the engagement when we are doing the stimulation. To be able to run that, we realized that there is no consensus in the field in terms of how to actually run these trials in the best possible quality. So we put together something like a methodological checklist that is being published in Nature Protocols right now. And that is the kind of the pathway that we are using right now in terms of how to run a frontoparietal, optimized frontoparietal stimulation that we have been discussing on F4 and CT6 with optimized level of electricity while we are exposing people to drug relative Q. And this is an ongoing trial that I already have that we are basically recruiting people on that trial. And I will show you at the end of my presentation how even we can move one step ahead in terms of not only recording while we are doing stimulation, but also optimizing the stimulation based on the recording that we are doing inside the scanner. And that is fantastic to see how you can actually do the stimulation inside the scanner, record from your kind of the effect of the stimulation, and optimize your stimulation. I will discuss about how that could be another level of optimization. Okay, so we discussed about frontoparietal network as well as the networks that we try to engage. The other network or the other area that people have frequently tried to engage was just DLPFC, trying to put electricity in DLPFC, trying to engage DLPFC. I'm going to stay here, so just making sure I receive feedback in terms of, okay, just, yeah. There is a director behind the scene just giving me instruction in terms of how to move. So, yeah, somebody else is in charge. So, this is DLPFC. I'm a person that I usually kind of like to walk when I talk, but yeah, sorry. So I try to kind of stay here. DLPFC, when you engage DLPFC, you'd want to put electricity on DLPFC. This is one of the first studies that we did when I was a kind of a PhD student doing electrical stimulation for methamphetamine use disorder. And putting electrodes on the right, anodal on the right side, cathodal on the left side, and injecting electrical current inside to be able to modulate drug craving. And we showed on that study that we actually can modulate drug craving if it is not a big pain for others. This is, I mean, the initial study that we showed that, oh, yes, we can change the drug craving that we actually do the electrical current. And we started to look into kind of how it actually works. And one thing that we realized was just, sure, very close to those things. Yeah, basically F3, F4, putting 2 milliamp electricity for 20 kind of minutes. That is the kind of the more traditional way of doing PDCS in the first days. Now people are doing more innovative things, but yeah, that is very close to the initial kind of protocols. Putting the electrodes, injecting electrical current, and seeing that yes, we can change the craving experience. And interestingly, I mean, there are multiple studies that people have done. This is the kind of meta-analysis that we are doing. There are at least 24 trials with F4, F3, anodal on F4, cathodal on F3, and 14 trials in F3, F4. And they basically show that, yes, the craving could be engaged or modulated, and also consumption. Probably a stronger evidence for F4, F3, but meanwhile, the protocol that we have been using as well. But I mean, there are more studies as well on that group. So these are kind of something that people are doing in the field as well. But one thing that we realized recently, that when we actually put the electrodes on F3, F4, we are not really injecting current to DLPFC. When we put electrodes on these two sides, we're actually, the frontopolar area is the area that would receive the highest level of electricity. And this is something that we published discussing about. I mean, all those studies that people say, okay, we are actually stimulating DLPFC. You're not actually stimulating DLPFC. You're mainly stimulating frontopolar cortex. And that is the area that will receive the highest level of electricity. And then it got even more interesting when you see that people basically have different levels of, and this is a trial with 60 people with methamphetamine use disorder, okay? if you put electrodes on F3, P3, F3, F4, you can see the level of electricity in different individuals would be significantly different. And interestingly, we showed that in a kind of a preliminary assessment, the level of electricity in the frontopolar cortex would predict the level of response to a stimulation. And then we have been kind of working on that, and there was a new publication by Michael Fox and Sean and colleagues in Michael Fox lab in terms of their lesion study, the famous lesion study that's published in Nature Medicine. And the area that they show that there is a kind of significant connection, I mean, the connection of that, basically the connectivity network of that specific area would be related to change in behavior for substance use, whether it's nicotine or alcohol. And there's a kind of interesting story behind it as well. I mean, I was in a kind of, in a Saturday morning, I received an email from, I think, New York Times, and an interview in New York Times. I was thinking that's probably the sham, kind of a fake email. So who's kind of sending me an email on Saturday morning? I usually do not kind of respond. I respond to my emails on Sunday, but not very much on Saturday morning. So I was looking, and I kind of searched for the name of the guy, and I realized, yeah, he's a kind of a legitimate person. And I realized, okay, and I replied back in terms of, it seemed that there was a plan for a paper that was going to be published in Nature Medicine, and the paper was supposed to be published on Monday, and they have been struggling to understand the paper. It seemed that the editor asked to basically put some like an editorial or a specific report about that paper that's going to be published in Nature Medicine, and they have been just trying to find some way to help them to understand how the actual paper is written and what is the final message. So I was interested to the paper, and because I was interested to help them, I spent my weekend and I basically read the entire paper A to Z. And this is something that we usually do not do very frequently these days, of reading the paper from the kind of cover to cover, right? The entire paper and supplements and all the figure and other things. And I really enjoyed the paper, Sean. So it was just a fantastic paper, like many other papers that you wrote. Yeah. And I really enjoyed that. But when I was reading actually the paper, I realized, oh my goodness, the area that they are discussing is very close to the area that we have. It's very close to the frontopolar basically site that we say that is basically the area that the level of electrical current is predicting response to TDCS for a craving management. So I was excited. So I basically sent an email to Mark Fox and mentioned, okay, Mark, we have this data. Is this something that you would be interested to share? And he was interested. So he basically kind of shared his data. And I was looking into his data and I realized, oh my goodness, that area, we also have a really strong activation in drug cue reactivity maps that we have from methamphetamine users. So I was thinking, oh, we have activation of that area in our methamphetamine users as well. And I was thinking, okay, let's just see if we have the activation in this area in other areas, in other substance users. So I contacted Colleen and mentioned, Colleen, are you interested to share your nicotine and alcohol data? And Colleen shared her data. And I realized, oh my goodness, it's interesting that all of us, we have the same sort of pattern, not exactly the same, but similar pattern of activation in frontopolar area when we expose people to drug-related cues, which is close to what they showed in the lesion study. And they are overlapping to each other, which is very encouraging. So on the top of this, I mean, Sean and their team, they showed that if you do computational head modeling for people who are kind of doing the smoking with the trial that has received FDA approval for H4 coil for TMS, and they showed that basically the computational head model of that area, that coil, is very overlapping with the frontopolar cortex, and that is another supporting evidence. And also H7 coil that there are kind of few but strong pieces of evidence that potentially could be helpful for alcohol use disorder. That area would also be overlapping with the area that these coils are targeting. So, I mean, these are just converging pieces of evidence that is an important area for neuromodulation in the field of addiction. So, and there are also kind of studies for what Colleen Hanlon was targeting with her line of TMS on what she was calling as VMPFC or medial prefrontal cortex. computational health models of those areas are also overlapping with the lesion study. So we put all those pieces together, and that basically converging pieces of evidence ended up to the cover of American Journal of Psychiatry, February 2024. And that was a kind of an interesting kind of experience of having Sean, Mike, others, Colleen, everybody work together to just bring things together and kind of see how we would be able to bring our forces for something that would be helpful in the field of addiction in the future. But the idea of targeting VMPFC or frontopolar cortex and how frontopolar cortex stimulation could be an interesting area for neuromodulation for addiction, we have started to kind of extend that idea in terms of what could be the next level, how we might be able to use that idea to actually run a trial and extend that idea. We started to basically look into what we have been doing and also literature. There is a kind of really interesting study from Elliot Stein and Brian Adinoff team that they showed that one of the most important predictor of relapse in cocaine users is the engagement between an area that they called kind of VMPFC, left centromedial, the rostral anterior cingulate cortex. But it's the area that they are showing is actually mainly located on the frontopolar ventromedial prefrontal cortex and the interaction of that area to amygdala. So they showed that the interaction between these two areas was the most important predictor of relapse, okay? So when we had the actual map of cue reactivity on methamphetamine users that I showed you before, I considered that as something like a mask, and I measured the connectivity between that mask and entire brain in terms of the task-based connectivity, PPI connectivity with the entire brain. And there is a positive PPI with basically left centromedial amygdala and also right DLPFC. This one is positive, this one is negative, which is quite an interesting network to see how this specific frontopolar area is connected to centromedial amygdala and also DLPFC. So we have been thinking, okay, let's just kind of think further about how that network could be engaged and how we might be able to engage that network in the future trials. So we started thinking about, okay, if you want to run an individualized TMS trial, targeting that in that individual level, how we might be able to actually run that trial. So we started with a kind of a brilliant postdoc in my lab, Ghazale, who has been kind of working on this line of work for quite a while, and is the first author of the American Journal of Psychiatry paper that we put together. We basically started to look into the individual variation in terms of if we go for amygdala to frontopolar cortex in this kind of wider area of frontopolar cortex, what will be the variation of the areas that we have when we do individualized connectivity measurement? And we showed that, yes, there is a large variation between different individuals. It's something like 60 people with methamphetamine use disorder, X, Y, Z locations. So they could be distributed across different areas. And also, we showed that this individualized network or the individualized connectivity between amygdala and different areas of frontopolar cortex would also be related to the level of craving. And you know, it's not that much usual that you have a behavioral measure and an fMRI measure that are reasonably correlated to each other, and you can replicate that in different databases. So it is something that we have been seeing in our databases pretty frequently, which is pretty kind of encouraging. And we have started to develop a new protocol in terms of optimizing the location of the stimulation with TMS. And also realizing that how much the orientation of the coil, TMS coil, would make a difference in terms of the level of target engagement. So if you realize that when we play with the orientation of the coil, there will be a significant level of difference in terms of how we can engage a network inside the brain. And also moving for basically using robotic arms to be able to precisely kind of engage that network and do the right target with the right coil orientation. So that is the kind of the steep learning curve for us to learn how to use the robotic arm in the kind of in the right way, and also trying to optimize the dose based on the distance of the kind of the coil from the cortex, and also finding the best way to do the individual optimization in a way, making sure that at the end of the day,
in the trial, people who are receiving the same level of electrical dose in frontopolar cortex across different people in the trial. We try to compare how if we optimize the coil orientation, okay, based on the level of electrical current that we can have on that area. If we optimize that, or if we do not optimize that, basically comparing the fixed coil orientation compared to the time that we are basically optimizing the coil. So we show that if we optimize the coil orientation, we can increase the level of electricity that we can inject on that specific part of the brain. So the measure of optimization of engagement is the level of electricity that we have in the computational head models. The dose, and then optimize. So we have 3 levels of optimization. And we basically develop a specific power plan for that. And this power plan is published in Addiction Biology basically today. So I basically added this slide today to my slides. I'm happy that finally this is being published. But the entire kind of details and we automated the pipeline in terms of making sure that people come to, and then we finalized the trial. We had a fight with FDA, received approval, and now we are hopeful to recruit our first subject probably within the next 1-2 weeks. So we are kind of pushing that forward. So I'm happy that we already have the protocol in place. Please. And then, the idea that we are working on right now, it seems that based on the data that we have a sort of a dual target network in terms of we have a frontopolar cortex node which positively engages nucleus accumbens and amygdala together. And then DLPFC node, which negatively involved with that sort of cortical limbic network underneath. And it seems that the evidence is supporting that in terms of both targeting DLPFC and also frontopolar cortex could modulate that network. But the idea of potentially suppressing frontopolar cortex, stimulating DLPFC would be the augmentation. And there are already ongoing trials, and there are few, if you are interested, I can even give you further details in terms of there are already studies that they showed that if you do DLPFC stimulation, you have an effect. If you do frontopolar, you have an effect. If you do both, you can basically augment the effect of the stimulation, which is promising and interesting. The trial is still small, like 20 participants in each arm, but it seems like it's going to be an area for work for addiction. And what we are doing right now, we are trying to not only for frontopolar cortex, but also in DLPFs, and finding an individual network of what we are discussing about and how we can engage that. If that's an area for kind of that you are interested, I have some couple of slides at the end. If you are interested, I can go to the details in terms of we are always interested to see how much that network is also reliable. In terms of if I do it today and if I do it in 2 weeks, that specific individualized target is going to stay there compared to going from here to here and then from here. So how that specific network is going to stay there. So there are interesting things about that piece as well. Okay. So just last few slides to wrap up. Another area that we are interested and working on is, as I have mentioned, we are very interested to close the loop in terms of doing both recording and stimulation and optimizing that inside the scanner. And I'm really happy that we have been able to kind of solve some of the technical challenges of that with 2 brilliant postdocs they had, basically making the system work in terms of being able to record inside the scanner and also do the stimulation, and then see how stimulation is changing the recording, and based on the optimized kind of target that we have, trying to modify stimulation protocol. We basically published one of the first technical reports about that, and we have recently finished a trial with 20 healthy participants to show how we can actually do the engagement with this simplex optimization algorithm. But we are realizing probably there are some errors that we probably have made that we are trying to right now use what we call a Bayesian optimization. So we are optimizing that specific pipeline. So this is another area that I'm very interested to have a postdoc, and we are discussing and we are working on that specific line of work. So wrapping up what I've been mentioning. So we have been discussing what what could be the addiction targeting biomarker? What could be the area that we can target for addiction? We have been discussing about potentially multiple biomarkers that could be used for targeting, large inter-individual variations that we have, and how we might be able to partially optimize those targets, and also think about how we can do group level optimization versus individual level optimization, and also how we might be able to do dose optimization. I think I follow the literature in the field of neuromodulation for depression, and those who believe that individualization is going to be the final response, or those who think that, okay, let's just do the shotgun approach and just go for deep TMS and just kind of target different areas, and which one is going to be the best kind of area. And also kind of the recent discussion in terms of, okay, is this the DLPFC subgenual is going to be the best target, the most positive, most negative, or is actually there is some like a noise there as well regarding the respiration noise, how that could basically change the story. I think definitely individualization is an area for further exploration and discussion, but I think dose optimization also a really important area that sometimes is missing in the field. So I think that dose optimization is definitely an area for further discussion and optimization, and also what would be the predicting biomarker of response, what we are discussing about, and the challenges of personalized trials. As you probably know, when you want to run a personalized trial, you always have a challenge in terms of what would be the control condition in a way that you will be happy at the end of the day, and also the reviewers will be happy when you just have a sham kind of a stimulation compared to a kind of personalized one, or you have personalized versus non-personalized. So all those challenges for personalized trials that we have been discussing. Okay, last piece, just few minutes at the end. One thing that I'm realizing working with sort of the field of neuromodulation addiction, I'm realizing that it's not just us in the US who are basically making the main contributions to the field of addiction neuromodulation. There are other countries that are involved, and other labs around the world that are working in this area. So I realized it would be helpful to just have a network of labs that are doing addiction neuromodulation across the world. So I contacted many PIs across the world, and if they are interested to join a network of labs, and I have been able to bring them together in the first publication that we put together for International Network of TSTMS Trials for Addiction Medicine with Colin Hanlon, who was the main support behind that. And that basically paper recently, I was just checking it, it has some like a 300 citations so far. It's just a consensus paper in terms of how we can bring our forces together and work together in this area. And during the pandemic, we put together, that's the network. We already have 54 lab members across the world that are involved in this network. And during the pandemic, we put together another paper about how basically research in the field is being impacted by COVID, and how we can basically, all those trials are being interrupted, how we can just still run those trials and publish the results, and the guidelines that we probably can do. And we have a YouTube channel that we have something like a webinars, and we put the webinars on the YouTube channel so people can have access to the YouTube channel, and the videos are available there for the use. We had our last meeting in July in New York, the neuromodulation meeting with 28 speakers. We invited people from FDA, from NIDA, from the IEEE, different organizations discussing about what is going to be the next level for addiction neuromodulation. And this is something I really enjoy doing in terms of, I believe that science happens in a teamwork, and team science, not only within one institution, but also across different institutions, would be something that we can invest on. So this is a kind of a living systematic review, and I work with a couple of RAs and postdocs in my lab, and we try to keep it as some like a living systematic review about whatever that is happening in the field, to be able to track the changes and updates in the field. And the last slide, we are working on a few other networks in terms of also collecting data from different labs across the world. Labs that are using fMRI as an outcome measure and putting them together. Right now, we developed a network within the Enigma Addiction Working Group with Hugh Garvan and colleagues there, collecting addiction cue reactivity data. I was just looking to data. We already have over 1,000 participants with the fMRI drug Curac data already there in the database being processed. We are putting together kind of a new paper out of that. And we already have commitments for another something like 2,000 further subjects with drug Curie activity that they are going to join that database. And if we have larger database, we definitely can kind of do further exploration for the biomarkers that we are discussing about. And the last piece about, at the end of the day, whenever we are discussing about those things, I really like to make sure that the communities who are contributing to our research in the field of addiction or psychiatry also inform and are part of the innovations that we are doing. So I developed a network in Minnesota, a kind of Minnesota network of innovations in recovery, and we have all those recovery communities in Twin Cities, in Minneapolis and St. Paul. We have meetings with them, we get them engaged, we discuss about, okay, what does it mean to have neuromodulation for addiction? What is that? What we are actually doing? What are the questions that you have? If it is going to be something that you are going to use in the future, what are the considerations that you want to be considered in developing? So trying to have them as a part of the network or innovation network. Not only we are doing something for you guys to use, so just having them as a part of the activities that we do. For that specific line of work, we also kind of develop different sort of educational material with those communities and developing cartoons discussing about brain, why brain is important for recovery, why different kind of circuits in the brain is important in the process of recovery. And then colleagues around the world, they start to translate them in different languages. We already have them in 22 different languages, and they are being used across the world. And that is another sort of exciting thing that we do in the field. So thank you very much for your attention.